So now we have a basic idea that protein folding is energetically favorable. But we also know that the unfolded state can take on many, many conformations. So it could be reasonable to assume that the folded state could also take on multiple different conformations. But we know that a native protein will have typically just one native folded conformation. So this raises another fundamental question. How do proteins achieve that one single native fold? So to answer this, we're going to turn to a classic experiment by Christian Enfinson, one of the famous early protein biochemists. To do his experiments, Christian Enfinson used RNase A. And then there's a story behind why he used RNase A. It turns out that the armor packing company of hot dog fame had purified a whole kilogram of this protein and was distributing small samples to interested scientists. So because of that, RNase has become a classic model for studying protein folding. So another reason that RNase A is very useful is that it's a nuclease, which means that it's actually relatively easy to assay its activity. So RNase A is a 124 residue protein that contains a mixture of alpha helices and beta sheets and 4 disulfide bonds. The disulfide bonds are pictured here in yellow. So Enfinson, before he got started, got a baseline information, which he just measured the activity of his protein sample and set that as 100% protein activity. So after measuring the baseline activity, Enfinson added 8 mol urea to his sample, and that's illustrated here as sort of the gray shading. And when he measured the activity in the presence of urea, he found 0% activity, and that's because urea unfolds the proteins into sort of random coils, random conformations. And then he removed urea using a technique called dialysis. And once the urea was completely removed, he measured the activity again and found about 100% activity. So the activity had returned, suggesting that the protein had once again refolded into its native conformation. But he didn't stop there. So thinking of those disulfide bonds I told you about, he repeated the experiment with one major difference. So when he added the 8 molar urea, he also added a reducing agent called beta-mercaptoethanol. So what beta-mercaptoethanol does is that it reduces the disulfide bonds to release the 2 free cysteines. So now there's going to be 8 free cysteines on each RNase A chain. And then he performed a dialysis again, which removed both the urea and the mercaptoethanol. And in the oxidizing environment of the test tube that he had that was just open to the atmosphere, the disulfide bonds could now reform. So after all the urea was removed, he measured the activity again and found about 90% activity, which meant that the protein had again taken on its native structure. So even though he could reduce the disulfide bonds, they could reform again into the native conformation without destroying the protein. Enfinson then performed a great control experiment. Again, he denatured the protein and reduced all of its disulfide bonds. But then, before dialyzing the protein, he added an oxidizing agent. So this neutralized the mercaptoethanol and led to reforming of the disulfide bonds in the denatured protein. Then, when Enfinson removed the urea by dialysis, he found that the protein was only 1% active. Why is this? He hypothesized that disulfide bonds formed at random within the protein when it was denatured. Most of these bonds formed between the wrong pairs of cysteines. When the urea was removed, then the protein could no longer fold back into the native state because it was constrained by these errant disulfide bonds. Using some back-of-the-envelope math, we can calculate that the correct combination of 4 disulfide bonds needed to form active RNase A would form by chance about 1% of the time. And that matches quite well the results that Enfinson obtained when he obtained about 1% activity in his control experiment. And then there's a part 2 to this control, and that's Enfinson's final insight. So he took this RNase A that now had the mismatched disulfides, or he hypothesized it had, and it only had 1% activity. And then he added to this solution just a small amount of beta-mercaptoethanol. And what this small amount of beta-mercaptoethanol will do is it will allow disulfide bonds to both break and reform. And so they can exchange to different cysteines, and there's a chance of the disulfide bonds forming the correct cysteine pairings. After he waited long enough, and by long enough I mean about 10 hours, he found that he could regain a lot of the RNase activity so that the sample regained as much as 90% of the original activity. And that suggests that the protein, with time, can again take on its native conformation and that it clearly wasn't destroyed. So to summarize the result of Enfinson's critical control experiment, the RNase A protein could not reach its native fold after the disulfide bonds were allowed to form at random in the unfolded peptide chain in the presence of urea. However, when the urea and the reducing agent were removed at the same time, almost all the protein was active. And this suggested that the native fold of the protein placed the correct cysteine residues in close proximity, and then the correct disulfide bonds then formed and stabilized the structure and activity of the RNase A.
Can you explain how urea denatures proteins?
So the answer actually goes back to the previous video. Urea is a very polar molecule. So when it's at very high concentrations, such as 8 molar, it will alter the hydrogen bonding patterns of water. And that will decrease the impact of the hydrophobic effect we talked about that will drive a lot of the protein folding. And so reducing the hydrophobic effect actually shifts the equilibrium of protein folding back towards the unfolded state. So that's why 8 molar urea will lead to unfolding of proteins. From the results of his series of experiments, Enfinson formulated an important concept called the thermodynamic hypothesis. And the thermodynamic hypothesis states that the most stable thermodynamic conformation of a protein is its native fold. And that basically implies that folding is a spontaneous process in cells. Another important lesson from his experiments is that the information contained in the primary structure, or the sequence of a protein, is sufficient to dictate its three-dimensional fold. So the protein doesn't need any templates or any other instructions other than the presence of a favorable physiological-like buffer. So the thermodynamic hypothesis suggests that if our understanding of the energetic forces that drive protein folding are sufficiently sophisticated and precise, we could actually perhaps predict the structure of proteins just from knowing their sequence or primary structure. And actually, for some simple small proteins, that is the case. So here in yellow, I'm showing you the predicted structure of a small bacterial protein. And the structure was predicted using a powerful computational algorithm that includes a lot of information about the energetics of proteins. And after those predictions were finished, the structure was determined using X-ray crystallography. So here in gray is the experimental structure superimposed onto the predicted structure in yellow. And you can see that there's actually a pretty impressive agreement between the predicted and experimental structures because they look very similar and they superimpose nicely. And that actually says that there's strong independent support from these predictions about the thermodynamic hypothesis, because the fact that we can predict it means that we actually understand protein folding quite well. So when we think about the process of folding of proteins, we often illustrate it using a protein folding funnel, like the one shown here. And the protein folding funnel shows the thermodynamic landscape, so the energetic landscape of the folding process. On the x-axis is conformational space, and that sort of illustrates all the many different conformations that the protein can take. In some sense, you could illustrate that with a large multi-dimensional space, one dimension for each of the dihedral angles that can freely rotate in the backbone, but that would just be impossible to show. So we just simplify it by saying conformational space is the x dimension. And then in the y dimension, we have the free energy. So then if you imagine that the protein starts at a high energy level of the unfolded state, and then it will slowly travel down the funnel towards its folded conformation, its native structure. And the native structure will be at the minimum of that energy landscape, at the minimum of the funnel, and that's the key of the thermodynamic hypothesis. Now, in some cases, on some of the paths, the protein might encounter a thermodynamic trap, like the one that's shown here, where there's a semi-stable folded intermediate on the way towards the native state, the most stable state. The protein folding funnels can actually take on many different shapes for different proteins. So each primary structure or sequence will have its own folding funnel. So in this smooth funnel case, the protein will simply start in an unfolded state and rapidly reach its native conformation at the minimum. So in this second example, there's a large flat region at the top of the graph where there's high energy. So that means that there are many conformations in the unfolded state that have about the same amount of energy, of high energy. And so the protein will sample many of these conformations before it reaches the point where the funnel suddenly drops. And so the protein will then fold rapidly in its native state after having sampled many unfolded conformations. In this third scenario, a protein might need to pass through a high energy intermediate before folding, and that's indicated here by the shaded regions on this blue plot. But many proteins probably have a rough folding funnel, like the first funnel we discussed, which is shown here in yellow again. And it has these thermodynamic traps that we discussed with semi-stable folding intermediates. The thermodynamic landscape or folding funnel of each protein is likely to be unique. As you might imagine, the shape of the funnel influences the kinetics of the process, or how long a protein will take to fold. So now we know that protein structure is determined by the primary sequence. And that's a good thing for cells, because life is complicated enough. So because the protein sequence dictates the three-dimensional shape, it allows the protein to fold into the right shape almost every time without any other templates or informations coming from the cellular components. But those protein folding funnels that we just looked at also hint at another important concept in protein folding, and that's the kinetics of protein folding.